straight through those. One block.
Okay, so we will get started here because, well, first off, we'll do the pre-introduction, which is welcome to one of 17 senior experiences. And then I want to point out that Julia and Nico will be talking tomorrow in this same room starting at 3:30 and then 4:10, talking about air quality. And so I want to invite you guys to come to that. And then next week we have another 8 senior experience presentations. And Charles, where are you? There. Charles and Emily will be anchoring that next Friday.
Don't go that far.
Huh?
Don't go that far.
Okay, so you're 16 and you're 17, right? So you're anchoring this relay. And so check the university calendar to come and see their talks as well. They'll all be right in here. Also, invited to the chemistry picnic, which is right after this. All you have to do is go out the building, go through Main Hall, straight across the street, don't get hit by a car, and in City Park. So we've got that all organized. It looks like the organizers are here to eat as well. And I want to welcome the fencing team. I haven't met you guys, I've just heard about you, right? Anything she told you is not true, okay? And I'll give you the same, not true. Anyhow, okay, with that all, and we want to welcome the family as well. With that all said, I want to introduce Kaylee. And that Kaylee and then Greg, just before her, Greg graduated last year. I switched research projects for the first time in 20 years. And it's a scary thing to go from viruses to yeast because, the research is infectious for both of them. And very enjoyable, but it is a tremendous challenge to, everybody's slowly getting that joke. It's nice how it works its way through the crowd. It's quite a challenge to change research projects, and that it takes a tremendous amount of patience. Kaylee has demonstrated that tremendous amount of patience in making things work. When we absolutely had no idea what we were doing, never used yeast except to drink beer with in my life, and same thing was true for her. We finally, slugged that out. And I'm not quite sure why she decided to work with me after having classes with me, but it's just the way it goes. Anyhow, very proud of her and that the entire research group is here to have them attend. We worked really hard to make things go. And I also want to invite you to BioFest. Hikari will be presenting her poster on yeast hybrids tomorrow. So if you want to do a little beer sniffing, if you're underage, or tasting if you're overage, please come and see that as an additional research project to the project at hand. Without further ado, I will turn it over.
Thank you all for coming. A lot of people. My name is Kaylee. I hope you know that. And I'll be talking about avoid the amyloid, elucidating the effects of prions in yeast during the fermentation process. So the first disease that people denoted as a prion-related disease is Kuru. And Kuru is transmitted via ritualistic cannibalism. And that means that people eat the tissues and the brains of their ancestors. And it reached epidemic levels in the 1950s and 60s in the poor people in the highlands of New Guinea. And it's a neurodegenerative disease. So this means that it causes dementia and loss of motor control, and it results from the formation of plaques and can lead to neuronal death. You can see some of these plaques there. There's a lot more plaques in the Kuru brain than in the normal brain. So I'm assuming that most of you do not eat your grandmother's brains. So it's probably unlikely that you're going to get Kuru. However, there are a lot of other diseases that are a bit more relevant. So these diseases are called protein aggregation diseases. And that's because this is kind of like an umbrella term under which prion diseases fit. So some of these diseases include Creutzfeldt-Jakob disease, Kuru, Alzheimer's, Parkinson's, Huntington's, and then in animals there's chronic wasting disease in deer, scabies in lamb, and then there's bovine, spongiform, and encephalopathy, which is mad cow. So the bolded diseases are all transmissible species to species, and so this means that if you have beef from a cow that has mad cow, then you can develop Creutzfeldt-Jakob disease. And so all of the bolded terms are considered prion-related diseases. So a prion It's just a protein that misfolds. And so it can misfold either by different types of stress, like hypoxia or lack of oxygen, heavy metals, or trauma. And then there's a commonly accepted protein-only hypothesis. And so that's basically just saying that most people do not think that any nucleic acid is involved in the transformation from a normally folded protein into its prion form. So Stanley Krusner won the Nobel Prize in 1997 elucidating a prion propagation hypothesis. That's basically saying that normally folded protein, when it is exposed to stress like hypoxia or lack of oxygen, it spontaneously misfolds into a prion. And so when that prion gets in contact with a normally folded protein, it causes that protein to misfold. And so this kind of spirals out of control and leads to what's called an aggregate. And so once a lot of aggregates form, and generally in the brain, if it's in a human or in an animal, then it forms amyloid plaques. And so these plaques prevent synaptic function, and so it leads to neurodegeneration. So basically, we know that prions are proteins that misfold, and they're important in a lot of diseases, so it's really important to study. But how do we do that? So we could use certain models like sheep and study rabies, but that takes at least 2 years to develop and for the disease to run its course, and it's generally really challenging to diagnose beforehand. We can also study humans, so we could use a brain from an Alzheimer's patient, but again, that disease can take years, 20, 30 years to develop, and it would be expensive and legally challenging to get the tissues of a human. So, how do we study disease that takes years to develop? Well, we use yeast. So yeast is a really wonderful model organism because it's eukaryotic, just like us. And it's been well characterized, which means that the genome for a lot of different strains and species have been sequenced already. And it's cheap, which is really convenient for Lawrence. It's genetically manipulated, which is exciting, and it has a fast generation time, which is really nice for the lab use. So, because I'm from Wisconsin, I'm born and raised in Wisconsin, the first thing that I think of when I think of yeast is the brewing process. So you may be thinking, Kaylee, you're crazy. Well, bear with me. So when we look at the brewing process, we start off with mashing, and that's where the sugars are extracted. And then it goes to laundering, and that's where the grains are removed. And then the mixture is boiled to sterilize, and then the hops are added. And so then this is the exciting part. You've got the yeast, the fermentation yeast, and so then that is put into a fermenter. So that fermenter contains what we call wort, and wort is just essentially a lot of different sugars that the yeast ferments. And so it does so in an anaerobic condition, so it has no oxygen involved. So it is not, it would not take a great leap to hypothesize that these fermentation yeast have prions because we're in this stressful environment so often. Okay. So this takes us into translational brewing. So that's where you take a couple commonalities like stress and prions that are related to both the brewing process and disease, and you study these commonalities through the brewing process. It's a bit easier, and we do that to ideally learn more about the disease. So just a couple terms to go over involving translational brewing. There's flocculation, and that's the clumping together of brewing yeast after the sugar in the wort has been fermented. Then attenuation is just how much sugar has been converted into alcohol. And then generation, in the laboratory, basically it's just 1 hour, and that's how long it takes one yeast cell to divide into 2. And then for brewing, that's approximately 3 days. So as I mentioned in the brewing process, yeast is thrown into the fermenter, and so it's thrown in there for 3 days as the yeast ferments the wort. And then after that 3 days, it's either flocculated to the bottom or to the top, and then they take out the yeast, and they throw it, they repitch it into another container of wort. And so one generation is how long that specific yeast is in one batch of wort. So one of the most important things that brewers look for in their beer, obviously, is the flavor. So that is due to a myriad of factors, including the energy that it takes to make flavor compounds. So you have a high mitochondrial memory potential, and the mitochondrial memory potential, I'll explain a little bit more detail. But that's just talking about the number of protons in the intramembrane space. So because there's a lot of protons, that means that NADH has been converted into NAD+. And so NADH is also involved in making flavor compounds. So because there's less NADH, that means there's less flavor compounds. And then also the aging of the yeast is important, because if the yeast are too old and they've been stressed out for too long, then the flavor of the beer decreases. So basically we can conclude that the mitochondrial membrane potential and the aging of the yeast is important for the flavor of the beer. This is convenient because it's also important in aging. Because in Parkinson's disease, one of the quintessential characteristics of this disease is mitochondrial dysfunction. However, they're not exactly sure what causes this mitochondrial dysfunction. And then Huntington's, Parkinson's, and Alzheimer's, all protein aggregation diseases, all occur later in life. And so this is due in part because the proteins recover less effectively to aggregation. And this can be because there are less chaperones or heat shock proteins that prevent the protein from misfolding. And then aggregations just simply occur and accumulate over time. So there are 9 known prions in yeast, but I'll be focusing on 2 of them, the sub-35 protein that turns into the PSI positive prion, and the MOT3 protein that turns into the MOT positive prion. So I chose these in part because they're very well characterized in the literature, but they're also important in the cell. Sub-35 is involved in translational termination, and so when that protein misfolds into a prion, then the translational termination activities are compromised, and the cells terminate translation less effectively at nonsense codons. So that means that a lot of proteins are made that really should not have been made. And MOT3 is a transcription factor, which just means that it's a protein that binds to DNA that regulates transcription, and it represses the hypoxic genes that regulate, that are under aerobic conditions. They have not done a lot of research on the function of the MOT positive prion. I looked at 3 different yeast strains in my study. They're all from Saccharomyces cerevisiae. And so the fermentation yeast that I used were 720 and 099, which I'll explain a little bit later. And then the laboratory strain I used was 359. So I chose 099 because it's super high gravity ale, and that means that it can ferment up to 25%, and it has a high attenuation rate of about 80%. And it originated from England, in fact. And then the 720 is kind of on that opposite end of the scale, so it's a sweetening yeast, and this means that it has an apparent attenuation of only 75%, and that it's known for leaving some residual sweetness and for being slightly fruity. And this is most probably due to the fact that a lot of the sugars in the wort have not been fermented into alcohol. And then for the laboratory strain, the 359, it's meaning type A, no meaning type switching, and it's been genetically modified in a variety of ways. Okay, so my study, what I actually did, I attempted to determine the presence of specific prions in the fermentation yeast that I looked at, that I wanted to compare the presence of these prions in specific generations. And I also wanted to determine the effects of these prions on mitochondrial membrane potential, aging, and attenuation. And so in order to do this, I needed to develop some sort of control in order to be able to compare between something that shouldn't have prions and something that does, so I know that that fact is in fact due to prions. So we used 1-didymium hydrochloride. So that's a compound that's been known to cure the yeast of prions. And so that basically means that it's preventing the hydrophobic interactions between the misfolded proteins so they do not aggregate together. However, the exact mechanism for how this compound actually cures the yeast is still unknown, but most people believe that it's due to the prevention of fragmentation of existing prion units. And papers have speculated that it's been in conjunction with other heat shock proteins. So my research involved a lot of use of picobreweries, as we termed. And so that's a 100 milliliter valve bottom flask, and that contains 75 milliliters of the wort. And wort, as I mentioned, was just a bunch of sugars that has to be fermented. So we obtained this wort from Stone Arch Brewery, and then we added 1 milliliter of whatever yeast we were studying for that pico brewery. And then for half samples, I added the guanidinium hydrochloride to form as a control. And the note air was allowed into the flask, in part to stimulate the brewing process, and another to make sure that the yeast was stressed out enough for them to theoretically develop prions. So my process was, I started with the pico breweries and I allowed the yeast to grow. And then I used the flow cytometer, which is an instrument that allowed me to analyze the aging and the mitochondrial membrane potential. And then I used a type of Western blot called SVDH, and that was an attempt to determine the presence of prions in those specific yeast. And then lastly, I used a densometer to measure the attenuation. So the flow cytometer works as a cell counting instrument, and so the samples are placed up there, and they go down this column one by one, and then they are shown through with a laser, and that laser serves to provide us a lot of information, but it mainly serves, in my case, to look at the type of fluorescence that it detected, because attached to all of my cells were different types of fluorophores, or fluorescent molecules. So specifically what I did to measure the aging and the mitochondrial membrane potential is to collect and dilute my samples from the pico breweries, and then I added propydium iodide, which is a type of fluorophore. And then for half my samples, I added rhodamine 123 to look at the mitochondrial membrane potential, and then the other half I added wheat germ agglutinin, and that was to look at the aging. So I specifically added the propydium iodide to all of the samples because that fluorophore intercalates the DNA, and so it only fluoresces the dead cells. So that way it's allowed to only have alive cells in my count for my data. And then rhodamine 123 is a fluorophore that is pH sensitive, and so that means that in this diagram you can see this is electron transport chain, and so there's a bunch of different complexes down here, and at the end it makes ATP, and it does so using this proton gradient right here. And so, therefore, the more hydrogens in the intramembrane space, the more ATP is able to be produced. And so what the rhodamine 123 does is it binds to the protons and it defluoresces. Therefore, the higher the mitochondrial membrane potential, the lower the fluorescence is going to be. Wheat germ agglutinin is a fluorophore that I use to look at aging, and I did so because every time the yeast cell divides, it leaves behind a bud scar. Right there. And so the wheat germ agglutinin binds to a bud scar. So therefore, when you have a lot of yeast with a lot of bud scars, you're going to have a high fluorescence. So that signifies that the yeast are older. This is just a graph that I would see on my flow cytometer for every sample that I took. And so this graph is looking at their fluorescence with size and complexity, and each little dot you see there is one cell, and I measured approximately 30,000 yeast cells per sample. And so this graph got turned into a histogram over here, and that is measuring the geometric mean. So as you can see on the bottom, this is in logarithmic scale. And so going forward, I will be talking about the geometric mean, and that's the average fluorescence of that sample. So getting into my data, I'll first talk about the aging. So these first three graphs are going to be the samples that I added the wheat germ agglutinin to. And so as you can see, with time on the x-axis and on the y-axis you have geometric mean, control in the red, and the wheat germ agglutinin, the guanidinium hydrochloride treated samples are in blue. So there is no apparent difference between these two samples, so we conclude that prions likely do not affect aging in the yeast strain 720. So very similar results were seen for 099. We concluded that there is very little difference between the control and the guanidinium hydrochloride treated samples. And so the prions likely do not affect aging in 099. And so these two yeasts are both the fermentation yeast, just keep in mind. Because for 359, the laboratory strain, it appears quite different. So you have the control in red up here. That has a much higher geometric mean compared to the control. And so therefore we conclude that the guanidinium hydrochloride treated samples slightly younger than the control for the yeast strain 359. So just to recap, for the aging, 720-099 fermentation yeast show very little difference between the guanidinium hydrochloride treated samples and the control. But for 359, the laboratory strain, the guanidinium hydrochloride treated samples are younger than the control for the yeast strain. So now we're getting into the effects of mitochondrial memory potential, and this means that these samples were treated with the rhodamine 1,2,3,4,4. And so, as you can see, the guanidinium hydrochloride treated samples in the blue is higher on average compared to the control. And so that means that because they have a higher geometric mean, that the fluorescence, that the mitochondrial memory is lower compared to the control. The same was seen for 099. The guanidinium hydrochloride treated samples are higher, have a higher geometric mean compared to the control. And so this suggests that for 099, the guanidinium hydrochloride treated samples have a lower mitochondrial memory potential compared to the control. So again, these are the first two fermentation yeast, and they all both appear to have that same pattern. So when we get to the 359 laboratory strain, it is very evident that that does not follow the same pattern, because the control in red is much higher geometric mean compared to the guanidinium hydrochloride treated samples. So we can conclude that the guanidinium hydrochloride treated samples have a higher geometric mean compared to the control. So just to recap, we know that 720 and 099 have a higher geometric mean for the guanidinium hydrochloride treated samples compared to the control. And so we suggest that prions likely positively affect mitochondrial memory potential for these fermentation yeast. So 359 did not follow these same patterns. So as I mentioned originally, I did a type of Western blot called SDDH, and it's semi-denaturing, semi-denaturing detergent agarose gel electrophoresis. That is too much of a mouthful for me to say ever again. That will be termed SDDH from now on. So that just means that, so the agarose here is a type of gel that I used, and so gel electrophoresis is where we run samples down a gel to separate them, generally based on size. And so the first thing that I did for these samples was to isolate the yeast from my cucurbers again. So then I de-beat them, and I did that so I could break open the cells so the proteins could get out into the sample. And then I separated these proteins by gel electrophoresis. I ran them down a gel in an attempt to separate them by their molecular weight or their size. And then I conducted Western blot, and that means that I attached fluorescent molecules to the proteins of interest, so I could image them, and then I imaged them. So just to remind you, the different proteins that I'm looking at are the sub-35 protein and the NOT3 protein. Sub-35, just remember, is involved in translational termination, and NOT3 is a transcription factor. And so we would expect to see, if their prions are present, that because there is so much aggregation involved in prions, that we're going to see a smear instead of a nice clear line like you would normally assume when you saw a Western blot. So getting into my actual data, this is an image of one of the SGD agents that I ran. And this is looking at the first generation, and I added the fluorescent molecule to look for the sub-35 protein. And so because you can see the smear for 720, we are hypothesizing that the yeast strain 720 does indeed have this prion. So getting on to the generation 2, you can still see the evidence for prions here for 720, but there still does not appear to be very much evidence for 359 or 099 to have this prion. So we're getting into the third generation. There still is evidence for 720 having this prion, but it's kind of exciting because you can see in the guanidinium hydrochloride treated samples there was no prions, and that's what we would expect, that the guanidinium hydrochloride, here the yeast are prions, so there would be no prions in that sample. 359 is showing a little bit of evidence for prions, but not a lot, and then 099 is still showing no evidence. Now getting into generation 4, 720 still has evidence for these prions, but 359 and 099 are not showing it. So for generation 5, you can see that 720 still has the presence of prions throughout the generations, and 099 is developing just a little bit at the end, but 359 has none. So just to recap, we conclude that the yeast strain 720 does indeed have this specific prion, PSI positive. There's very little generational differences, except we did note that for at least one of the generations, the guanidinium hydrochloride did in fact do its job and cured that table of prions. However, we concluded for both 359 and 099 that there was no evidence for this prion in either of those yeast strains. So now we're going to get into MOT3, and so that was looking at the MOT3 protein. So you can see here that for generation 1, 720 does indeed have this prion as well. And then for generation 2, there appears to be a smaller or lower concentration of this prion in 720, and again, 359 and 099 show no evidence for this prion. So now we're getting to generation 3, and as you can see, there's just a wee little bit of this prion in the 720, but again, for 359 and 099, there is no evidence for this prion. And for generation 4, there's no evidence for any of these yeast strains having this prion, and same can be said for generation 5. So just to recap, the yeast strain 720 has some evidence for the MOT3, and there were some generational differences in that the presence of MOT3 decreased over time, and both 359 and 099 did not show much evidence for this prion. So lastly, I looked at specific gravity, and I did this to try to measure the attenuation of the yeast. And I did this using a densometer, and that measures a specific density compared to water, in this case. And so the higher value indicates less alcohol has been made. So we're getting in here, you can see wort here, that's just the original value, and so that's 1.04. And you can see that 099 and for the control and the guanidinium hydrochloride treated samples, there is very little difference for the densities. So that means that the guanidinium hydrochloride treated samples, or prions, essentially do not affect the attenuation rate of this yeast. And then for 359, you can see that is still true as well. There is very little change in the densities between the control and the guanidinium hydrochloride treated samples. However, it's exciting because 720, the control has a much higher density compared to the guanidinium hydrochloride treated samples. And so we can conclude that the yeast produce more alcohol in the presence of guanidinium. Again, just to recap, for the yeast strain 720, the yeast are able to produce more alcohol in the presence of guanidinium. However, for 099 and 359, the production of alcohol is not affected by the presence of guanidinium. So some overarching conclusions that I have. Prions likely do not affect aging. And we can conclude that the prions PSI positive and MOT3 are likely positively affecting the mitochondrial membrane potential for 720 and 099. And so this is interesting because it has been hypothesized that for Parkinson's, one of the reasons why the mitochondria dysfunction is because the mitochondria are overactive in the beginning, and so they kind of tire themselves out, and so they stop being functional. And then also, because there's that increased mitochondrial membrane potential, there's also an increase in oxidative stress, which can also be damaging to the cell. And so these yeast strains would be an interesting choice to study prion diseases because of this effect that we saw. And then we noted that 359 does not follow the same patterns as fermentation yeast, and so it would be ideal if we could use both laboratory strains and fermentation strains in the future to study prions because they are so diverse. So challenges that I faced were that the guanidinium could have been affecting other things other than the prions. Because they have not elucidated the full mechanism for how the guanidinium cures the yeast of prions, it's possible that the guanidinium could be affecting something else. And the antibody Fermat-3 could have been dysfunctional. We were unable to obtain a control for this protein and prion. And then 099 and 359 could have had different prions other than the ones that I looked at. And I think that this is probably likely because we saw such a difference in the control versus the guanidinium hydrochloride treated samples in the flow cytometry data. However, I was not able to look at all 9. So some future work is to look at some other yeast strains and some other proteins and different prions in an attempt to understand the underlying mechanism for prions and how they function. And then other people, including Hikari, are doing work on hybrids. So they're hybridizing 2 different yeast strains into one. And so it'll be interesting to see how those hybrids deal with and if they have prions. So for to acknowledge Dave for all the work that he's done helping me in this process, and then Stephen Deverett for getting me through college. I would like to also thank Elizabeth Craig and Anita Morgan, something right. They both helped me in figuring out how to do the SBB age. It did not work for 6 months or so. So their advice and their protocols were very welcome. I would also like to thank Daniel Martin for supplying my ever constant need for material in lab. And then also my lab mates for getting me through my lab work and college life in general. Sources.
Question?
Yeah.
This is more about your methods, but how do you even run a prion on a gel? You would assume it would be insoluble. Like, how did you?
So, we isolated the proteins in like each sample that we looked at, and then so we basically hoped that these yeast strains had that specific protein. It's possible that they didn't. And so if they did and they were prions, then we would expect to see that smear. But if they weren't prions and they did have that that protein, then we would expect to see that clear line for them being essentially like one unit protein instead of being that clump, that aggregate. Does that make sense?
Yeah, I was just wondering, like, when you made your samples, were they like soluble in the? I mean, would you use SDS?
Yes.
Were they soluble? I mean, why were they soluble?
They're pretty indestructible, so you can do a lot to them and it's fine. I'm not quite sure what you